Uh, sorry, my screen is way too big. Well, we're just going to deal with it. Hi, my name is Ernst Pulido. I am a master's student in SFSU. I'm also a research associate in UCSF in the Krogan lab, in also the Gladstone Institute. Basically, my role in both labs is to do protein-protein interaction. However, for this talk, I would like to focus on my journey from community college to the cell molecular biology research that I am right now doing. The picture that you see here right now is you have two viruses. One top is the herpes virus, or KSHV, which is Kaposi sarcoma herpes virus. One on the left is actually the SARS-CoV-2, which one of my projects is in. And the last one is basically the microtubulin. So this is one of the structures that I've been studying, which is pretty cool. And so we'll definitely explain later on. Or not. So the outline, just to get everybody into perspective, I want to show the intro and the growing pains of me growing up. It wasn't all roses and daisies, so it was definitely tough being a first generation and also a minority in growing up as in East Coast of America. Then, as I will talk about the internships, and I think it's very important to have, and the projects that I'm doing, starting with the microtubulins, and then going to the viral host complexes that I specialize in. So a little bit of introduction. Let's make this actually smaller. So I was born in Philippines, Quezon City, close to Manila. It was, I stayed there for 10 years. So when I was 10, I actually moved to Hawaii, to San Bay Area in Pacifica. I also moved to Hawaii, but it was just a short time. And after that, I moved to Virginia in close to Washington, DC. And so I've been coast to coast. And recently, I also been back to the Bay Area. Now, you ask yourself, why is there a baby picture right there? I asked my family to send me a photo, a childhood photo of myself, and all they gave me is baby pictures. So that is what you guys get. Growing up, I actually grew up in Broad Run High School. It wasn't the easiest. If you look over here, a diversity score of Broad Run was not as good as you see as opposed to California state average. However, this I think is still kind of wrong, as it, I think it was 95% Caucasian when I was in Broad Run High School when I was growing up. And so there was a lot of, there was not that much diversity. There was not a lot of people like I am Filipino. And it was tough because even the teachers didn't think that I was able to go to science. So there was no, even when I asked one of my teachers to give me a recommendation if I should go to college, he actually laughed at me and said, I don't think that's a good idea. And so that was ingrained in my mind in high school. So I didn't have a spectacular push to science early on. Even I think next slide. Even the guidance counselor thought that I should go to military instead of going to college, which to me I think it was wrong. But back then I think it was because they didn't think that I had the chops to go to college. But here we are. So I actually started working as a Taekwondo instructor when I was 15 to be able to help our family because we didn't have too much. We weren't able to provide so much, so I had to help out. And then after high school, in my guidance counsel's advice, I started to be an accountant. Technically, it's not an accountant. It was like a junior accountant, but they didn't pay me as an accountant. But I did work as an accountant. They thought that I was a cheaper alternative to accountants. So I guess it's all right. I made good money, but I was always passed up for raise because I didn't have the bachelor's degree. So I actually had a child. His name is Joseph. He's 2 years old in this picture. However, my partner got really sick, and so she was in the hospital in and out for 5 years. And so for the first 5 years, I actually took care of Joseph while going to school, while working, and that was tough. And so there was a point where I was unemployed because I wasn't able to take care of my partner and of my son. And so I decided to go back to California and try to figure out what to do. And I was at my lowest point. In fact, I just took any job, and the job that I got was the garbage man because no one wanted to hire someone that didn't have a degree or didn't have anyone that vouched for him. And so at the end of working, actually it's a recyclist, but I go through people's garbage. After that, at the end, I told myself, hey, I really need to do something because my son is growing up. He's going to see that I've been maybe not pursuing something that I want to do. And so I wanted to be a role model to my son. And so I went to DVC, and I was like you guys, a DVC employee. First, I was a financial aide. But after that, I actually worked with Maitri as a student worker. And so in the MESA, so I was the guys' to choose, which is cool now to see that other people are in as a student worker. And that's actually when I started getting my internship. This is my first internship, the Pumas program in the Gladstone Institute. I will talk about it more afterwards, but this is a point where I had an internship. However, I wasn't able to make ends meet. I was a single parent at that point. And so my partner left, she got better, she went. That's all right, I'll take her, our son. And I took 2 jobs while going to school. So it was tough, but it had to be done. And so after I became park ranger, I actually went to my second internship in CCMI. And this is where I believe I transferred. And so the second internship was detrimental to me because that's the point where I wanted to be a researcher. And finally, I transferred to SFSU. It was because after the CCMI internship, I got into Cal Poly in Pomona and also in SLO. However, since they were offering me a job in UCSF, I said, why not? Let's go to SFSU and become a researcher. And so at one point, I graduated at SFSU and, pursued and also worked as a researcher full time. And so I'm still currently employed there while doing my master's at SFSU. I wanted to do a research where it was my plan, and so I could be able to show, my PhD, a PhD program that I'll be able to do the same thing. And so that leads me to 2001. I'm still a researcher, and I'm also almost done with my master's. I think I have another year left, and so hopefully by the end of this year, I would know if I'm going to be able to go to a PhD program. And so during my time in DVC, this is actually the time where Maitri was she pushed me to get an internship. I think her exact words were, why not try for an internship? And she showed me Puma's program. And at first I was not very receptive, but I thought, why not let's take a chance, you know? So I got into the Puma's program in 2017. My mentor was Dr. Hashim Sheheta, and the lab was the Sanjabi lab, and we were studying the regulation of CD8 T cells by SPRAT1 and 2 molecules. It was a good internship. I learned a lot, and there was a lot of what's it called? Yeah, I learned a lot. It was a lot of networking. This is the time where I actually, you know, it was a tipping point in which I want to go to research or not. At this point, I also received the SACNAS Travel Scholarship, so I attended this conference in the same year. So it was a pretty cool year. After that, my second internship was with the Cancer Cell Map Initiative in UCSF between Dr. Agard and Dr. Krogan's lab. And two of my mentor was Dr. Moritz and Dr. Cake. And we were studying the interaction of casein kinase 1 delta and delta tubulin ring complex using integrative modeling to be able to find a structure for them. And so for now, the science. So I would like to start actually in something that we have studied in Bio 130, I believe, the central dogma of biology, in which we know that DNA is transcribed to RNA and RNA is translated to protein. I think about the DNA as the main book in which everything is made of. And so this cell is factory that I in my head, it has machinery inside of it. And this machinery is the proteins, and the proteins need, need to be made. And what's making these proteins is from the DNA instruction. So the DNA is then has the copies of, not copies, but the main source of what's supposed to be, how you're supposed to build the machines. And you copy that because you can't take it out from its office. So you copy it, and that's the RNA. So once you copy it, that's transcribed to a paper, which is the RNA. Then the RNA can go to the ribosomes to be able to make the protein. And it's translated to protein. And this is super important to biology. Now, this protein is also interesting to me because once you have a protein interacting with each other, actually makes a protein complex. And this protein complex then interacts with each other even more and makes an organelle. Now, it has some other stuff like, what is it called? Oops. No, not going that way. It has other stuff like fatty acids and all that. But the proteins are basically the machinery of the factory, the machinery which is able to do stuff for the cell. And this organelle, when you have multiple organelles, makes a cell. And the cell then, multiple cells makes a tissue. Multiple tissue makes an organ. And multiple organs makes the organ system. So anything, if you go all the way back, anything that is wrong with a protein complex or its protein, then you'll feel the effects all the way down to the organ system. So it's super important to learn and figure out why these proteins are interacting with each other and how they are affecting the whole organ system. Now, how do we learn about proteins? How do I learn about protein complexes? Is that I figure out their structure. And if we know the structure and we know how it looks like, where it's interacting, we could do a lot in making, for example, antivirals, and we could do vaccines. And so we could try to figure out how to heal someone that has a disease through figuring out what the protein complexes are. Sorry, if you do have a question, or if nothing makes sense, please stop me. I don't mind. I'm used to it. So you could just unmute yourself and say, "Hey, Ernst, I need more explanation," or else I'll just be talking all the time. So moving forward, this is my CCMI internship within the Agard and the Krogan lab. Like I said, it is the casein kinase 1 delta and the gamma tube. Do you have any questions? Cool. Gamma tubulin ring complex using integrative modeling. Now, integrative modeling is basically taking two or more parts of what's it called? Visualization of the proteins, and you'll be able to make a structure or a picture of the structure of the proteins. Like you can see over here on the right is the cryo-EM of gamma tubulin ring complex. If you're able to get, for example, Crosslink-Mass spec into this information, then you could see where those proteins actually interact and could stop those interactions. Now, I don't want to make it too complicated, so I'll just move on. Two of the mentors that I have in this project was Dr. Michelle Moritz and Dr. Robin Cake. So they've been detrimental into this project. So what is microtubules and the casein kinase? So the casein kinase on the left over here is the green lines over here. And so they're made out of alpha tubulin and beta tubulin. And they're important in meiotic cell division, as we know, in bio 130, transportation. It provides the structure of the cell, and it also provides other things and some stuff that we don't even know. And so this is the structure of the microtubulin. It's basically, like I said, alpha is the pink one, and the green one is beta. And then there's this protein that we didn't know in 2010 that actually makes possible for the microtubulin to nucleate or to build up upon. So in 2010, they were curious to see what that is, and they were actually figuring out that it's a ring complex. So this ring complex is holding the microtubules so that it could stay, in its shape. And so that is the microtubulin. The casein kinase delta, 1 delta, this is the protein for it. While it looks very, doesn't make sense, it's actually a kinase. So this kinase is basically what activates and deactivates proteins. So it goes around and it activates a protein to be able to do its work. In this case, the microtubulin, we saw that the ring complex in the bottom right here is activated by casein kinase 1 delta. And this actually activation is to close it. And sometimes when it comes off, it opens, and then it comes on, it closes it. So we want to figure out why does it close and open, and why, if it's closed, then you're able to nucleate or build upon the microtubules on top. But if it comes off, then it doesn't build on top of it, and it just stays open. And we wanted to know why is that. And so just a little bit more detail, because this probably makes sense, needs for later on. It's made out of two, let's move this on here, made out of two stems, which is the GCP2 and GCP3. While you don't need to know what that is. You do have the gamma tubulin on top of each, and this is where the alpha and beta tubulin starts to nucleate on top. These two stems is called the gamma tubulin small complex because they're smaller. And then the bigger complex is called the gamma tubulin ring complex because it looks like a ring. And like I said, it makes a ring, it opens up when it's not conformed. And then when you have casein kinase or other kinases that comes in, it actually closes it up. And so, and on top of that, we don't know where this casein kinase actually interacts. Does it interact with GCP2, or does it interact with GVCP3, or does it interact with gamma tubulin? We didn't know. But we know that this gamma tubulin ring complex is implicated with different things like cancer, with lack of sleep, with growth. And so we want to know if this casein kinase is controlled by cancer, for example, or is controlled by a virus or a disease. And so our integrative modeling is basically our workflow. First, we have to purify the proteins, and then we have to highly specialized experiments that the Krogan lab and the Agard lab does. This is actually high-powered machines, and not a lot of people could do this. That's why I thought it was really amazing. One of them is Crosslink-Mass Spec, and that's done in the Krogan lab. And then the other one is Electron Microscopy, which is done in the Agard lab, or Krogan lab, and yeah, this is the Agard lab. And so there's two high-powered machines giving us information. We take them together and then use that to make a structural determination, and that is called integrative modeling. And so I apologize for taking this slide from other people. I just didn't have time, but this is basically what we did in purification. You have gamma TURC, which is gamma tubulin ring complex, and casein kinase delta, and we have its DNA. And then we insert that into, for example, bacteria, or for gamma tubulin ring complex, we inserted that into insect cells. Then we let it grow so that we'll be able to have a bunch of it. And what actually happens is that this DNA that, for example, casein kinase 1 delta is transcribed to mRNA and then translated to protein. Again, this is the basics of biology. So this cell has the protein that we need. And what we do then is that we lyse the cell and basically destroy the cell wall. And so when it destroys the cell wall, you have all this stuff, which is the DNA, the proteins, and some, what was it called again? Mitochondria and all that stuff. I forget the word. And so you have all this stuff in this after lysing the cell. And so what we do is we actually fish out the proteins that we want to be able to purify and remove all the DNA that we don't need, all the proteins that we don't need, all the fatty acid that we don't need. We could just have the protein that we want. And this is our result showed. So this would be results. So HRR25 is actually gamma tubulin. No, it is casein kinase delta 1. And you can see that when we visualize this in the gel, we actually see the proteins in the right spot. And including what was called the gamma tubulin ring complex stuff, we could see it that we have purified. Well, there are stuff right here that we don't need, and that might have been detrimental in our experiment. But the more important part is that we do have GPC2 and GCPC3 and gamma tubulin, and gamma tubulin. And so we take this proteins and we put them together. We combine them together, and I fitted crosslinkers, not fitted crosslinkers. I use crosslinkers. It's like a chemical tape. Basically, this crosslinkers are specified to a amino acid. An amino acid makes the protein. So if it is specified to one amino acid, it will stick on it because it has a high affinity, or it just really likes that amino acid. And if that amino acid then is on the gamma TERC, for example, and if there was one in the casein kinase 1 delta, and there's a lysine close by, it sticks to it too. So you have both of them sticking to each other, and it has to be in a specified space, around 30 angstrom. So it's really, really small. And so between gamma tubulin casein kinase, we're able to tape this protein together and put them in the gel to separate out all the stuff we don't need, like you see right here. And then everything that is either cross-linked right here is going to stay on the gel. This one right here, I'm sorry, I forgot to put the labels on this gel, is no cross-linkers. And we could see that SPC, GPC, GCP 2 and 3 is right here. This is gamma tubulin, this is the casein kinase delta, and this is the gamma tubulin on top. So we are able to have those proteins together. Yes, Maitri.
Quick question, when you are separating the proteins, like you get a soup of proteins, right?
Mm-hmm. That is
and
true.
then you're trying to identify the proteins through by running the gel and seeing.
Yes. So one thing that I did not go over was that we used a column to fish out this protein. So these proteins have a high affinity to a certain column, and it sticks to it. Everything that doesn't like that column, just passes through it, and then we could discard it. And that's why you see it over here. All of the stuff that we don't need is right there. And so after cleaning it, you are able to just have just the protein that we need. Though there is this dirt that I don't know why it's there, but you do your best.
Thank you. That was my question.
Okay, no problem. And so this gel right here has cross-linked proteins. Basically what I do is I cut those gels and then I digest it. There should be something here. So the digestion is like, do you know the game, what's it called? Fruit, the one that you cut up the fruit. Basically, I take this protein, I use another digester or trypsin, and I cut up the proteins into a bunch of million pieces. And so it becomes just amino acids afterwards. And so you have the amino acids that is from gamma tubulin ring complex, for example, and you'll have amino acids that's just the casein kinase 1 delta. However, you'll have also proteins that has the cross-linker still attached to it that has the gamma tubulin ring complex and the casein kinase 1 delta. And that's what we hope to capture. This protein right here is what we hope to capture. These amino acid sequences we hope to capture. And this one right here, there are going to be some proteins that amino acid that's going to have the cross-linker of itself. And I think that's we're still interested in that. But the more important part is the one that shows the intersection between two proteins. And so once you have that, we actually feed it through the mass spec, and then the mass spec will read the amino acid and also where the cross-linker is, and then it will basically piece together like a puzzle and make a picture of the interaction between casein kinase and gamma tubulin ring complex, which I think it's cool because it's I don't think any other machines can do that. Oh, there it is. Fruit Ninja, that was what it was. So this is the result of our experiment. So this is called a circus plot. So this HRR25 I've labeled over here is casein kinase 1 delta. And you can see this actually interacts with SPC 110, which is the stem of the protein, TUB4, which is gamma tubulin, which is the top part of here, it also interacts with casein kinase 1 delta. More surprisingly is that this casein kinase 1 delta also interacts with SPC 97 and 98, which was cool because that's where we think that the casein kinase will be interacting. And then we take that information and we put it on the protein structures. And these protein structures then will show you where each amino acid is really close towards them. And so once you have that data, you can actually do a lot with it. Next one. And so the next part of this integrated modeling is electron microscope. Now, I didn't do much of this, but it was really cool to see how it's done. Basically, you stain the sample. So you get this very tiny plate, and then you use uranyl formate. And uranyl formate is radioactive. And so that is how you stain these proteins. And these proteins then is placed in the electron microscope, and then you see an image. And if the image is good enough, then you go to the cryo-EM to be able to freeze it to 170, I think 172 degrees Celsius, and basically stop the protein from moving around, and then shoots a EM to be able to see the structure of the protein that's frozen. Now, when I did this, I stopped at electron microscope because my samples was too, there was too much and I couldn't see anything. So I still have the image, but I think I left it out because I wasn't able to do the cryo-EM itself, which is still cool because I've seen someone else do it. So, and so this is the results of the long, hard work that we've done. You can see that HR25, which is casein kinase 1 delta, actually interacts more with GPC3, the right side. We do have MPV. It makes the casein kinase delta more stable, and we kind of think that we have to take it off eventually. So we're still trying to figure out how to cleave it off without making casein kinase delta just be not the protein that we need. And so we're able to, solve the structure by using the cryo-EM structure, which is the green part right here. And we know where the interaction is done by the casein kinase 1 delta. So future directions for that project. Actually, we are trying to publish it into a, so what happens in science is that once you have made a big enough discovery, you could publish this in a journal, and then this journal will be able to print out your articles and the world to be able to read. What happened with this particular project is that since SARS-CoV-2 happened, it's been shelved. So I think that will be, to be determined when that is going to be able to be, what's it called, be sent out. But it's still exciting because I know that since I'm being part of that project, I'll be able to be cited there too, but it just takes time. I've listed some of my projects in the lab here. I've done HIV proteins. I still have project with the HIV proteins. I do have projects with enterovirus proteins. That's also being pushed to be published. So hopefully, actually, we did send that out today, and I think it's being, we're seeing if it's gonna be accepted in Cell. So hopefully it gets in there. But the other proteins projects that I've been working on, I do have chlamydia proteins, influenza proteins, SARS-CoV-1 proteins, SARS-CoV-2 proteins, the CRISPR-Cas9 in the Dogma lab, which she won the Nobel Prize for in chemistry which she's a really cool person. The MERS proteins, proteins that are associated with neurodegenerative diseases like the BAG3, and then the KSHV proteins, which is the Kaposi sarcoma herpes virus. Thank you. So I'm hoping the ones that in red are projects that I'm working right now in my master's and also in as a research associate. And one of the projects that I want to share with you is I just got in today, so it was pretty cool. It's called the protein analysis of SARS-CoV-2. It's protein interaction between NSP2 and RAP1 and GDS1 interaction. This project is actually from the QBI. They are a big group in UCSF. And it's led by Dr. Meghna Gupta and Dr. Klimt Verba, and they took me in because I have, what's it called? I'm known to be the crosslinker in the lab, so to use crosslinking in the lab. And so, like the gel that we did, I just wanted to show we were successful in not only separating the proteins, but we also cross-linking the proteins. So I just want to go over this one real quick. This one right here, the top part is NSP2, which is the viral protein for SARS-CoV-2. And RAP1 GDS1, we actually don't know a lot of it. If you look at the literature, there's not that much things that we know about it. But we know that it interacts with each other because the cross-linkers work. What's amazing is that, I lost my arrows here, is that there's 3 different conformation protein structures that we're seeing. So we have one right here, which is close to 770. We have one on top of it, and then this big whole band. So there's 3 different structures that we're seeing. This is happening. This it's there. And so, like last time, we had the circus plot, and basically how you read this is that you have one side which is NSP2, and the line determines that this amino acid is interacting with the amino acid in the other side. And then the green line says that this amino acid is actually interacting with its own self, which means that this protein, RAP1, GDS1, is a little floppy. So it either goes down or goes up, our interactions. Sometimes it might curl. We don't know because we don't have a structure for it. But what's successful is that our false discovery rate is around 0.1%. So we're highly confident. We don't know if this is exact truth, but we're highly confident that there's interaction between these 2 proteins. And then we found 63 unique linkage between them, meaning that there's 63 points where NSP2 and RAP1 GDS1 is actually interacting. And so that was pretty cool. I just got the project today, so this is not a complete picture, so I would stay tuned with this project, which is exciting because if RAP1 GDS1 is important to SARS-CoV-2 and you take it out, then you have found a potential antiviral therapy that may happen for SARS-CoV-1 or maybe SARS-CoV-2, which is kind of exciting. And so my main project, I just want to go over real quick, is with a different lab. Dr. Sanchez has been my mentor on this project, which is the interaction between KSHV, KSHV and its potential role in hijacking the cell's metabolism. And both Dr. Cake and Dr. Sanchez has been helping me out making this project. And this is actually my project for the NIH, which is the NIH RISE Fellowship. So they're the one who's paying for this experiments. So I just want to, go back and remind everyone that, yeah, I didn't have the best starting position to be doing all this stuff. You would say like, oh, this guy didn't look like a researcher when he was 15, when he got out of high school. Being a first generation and also being a minority in a 95% school that's more Caucasian, it just it was stacked against me. However, I think this 4 is what you need to be able to get to a point, because I think everybody could do what I could do, but you just have to be able to one, have grit and perseverance, and then to be patient and to get an internship, and most importantly to have mentor. If I didn't have Meichi in my side, I don't know where I would be. So the other mentors that are listed here, I have them in highlighted. Michi, again, who has been an advocate for me in MESA, Dr. Robin Cake, and you saw that all my projects is with her. So she's also who believed in me and had a lot of patience. My new mentor, Erica Sanchez, Dr. Erica Sanchez, was basically helping me outside of just science. And Dr. Michelle Moritz, who her first project actually is the reason why I want to be in this field of biophysics, to be able to find two proteins that are interacting with each other and be able to see if we could build a antiviral therapy between them. Dr. Scott Roy and Dr. Eric Keene has been also detrimental to me, because Dr. Scott Roy has not only I have different projects with him that is totally different from what I'm doing right now, but he also pushed me to go for my master's degree. And so he has been detrimental to that. And Dr. Keene has been helping me with biochemistry, because biochemistry is hard. And here's my baby pictures again. If you have any questions, please send me an email at ernstpulido@gmail.com, or you can ask them right now. We do have a club that I'm starting in UCSF. It's called the IED, or the Infectious and Emerging Disease. And we meet every Friday, actually at 4 to 5. It's almost there. But basically what we want to do is to be able to share our science to between labs, because we don't know what we're doing. And I'm sure in community college, you guys don't know what other labs are doing. And so this is, I think, a good way to see, hey, do I want to be in science, or do I want to be in what's called in research? And so if you join this club, you'll be able to see that. We do have one do guest speakers too, like I'm doing right now. And it's not just going to be me, it's going to be someone that's, for example, a graduate student from UCSF. And if you want to meet someone from UCSF and UCSF is your dream school, that's a perfect way to meet them. I'm trying to get other people from other schools like Stanford, UC Berkeley. I think I do have a couple of people that wants to talk to us, and different places around the world. So if you guys are interested, please send me an email, or send an email at diseasejournalclub@gmail.com. Any questions?
Ernst, there was a question in the chat. If you could go back to the slide.
Yeah.
And talk more about, they phrased it as winning personality qualities.
Is it this one right here?
I think so.
Awesome. So yeah, I said grit and perseverance because you see, and when I started, there's always a lot of ups and downs. I mean, I went from taking care of a child for 5 years to be unemployed, to work as a garbage man, and then having people believe in me and actually go into internship, and then losing help, but then also finding what I want to do. Provides a lot of grit and perseverance, because that is very important. And then I also was patient, because everyone in my life at at some point didn't believe in me. My parents thought that I should go to work and not go to school. They thought that I should go to the army. In high school, they thought the same thing too. My partner left me because she also didn't believe in me, but I was patient, and I actually figured out that, I'll be able to take care of my son and be a researcher and be able to live in San Francisco, where I am right now. Actually, I live close to UCSF. And so it takes a lot of patience and perseverance. Other things that I think you do need is to get an internship, so if you want to be in that field. Without internships, then you'll feel like you go to that field and then there might be surprises that you never seen before. And that's something that internship might be able to provide. My first internship wasn't that great. It was terrible. One of my mentors was really negative, and I thought that I should know about biochemistry even in my first year of DVC. And I haven't taken organic chemistry at that point. But I learned that I don't want to be in that space. I want to be in a space where that helps me grow. And so, internships can provide that space. And then the last one is mentors, because, for example, without Maitri, I don't think I would be going to my first internship. In fact, I think I haven't helped other people get into the Pumas program, which is awesome. So if you want to get an internship, let me know. We could plan to get you into that internship. Did that answer the question? I know it's a little long-winded.
No, that was perfect.
Awesome.
And then feel free to put in the chat. Heidi was the one that had asked that question.
Yeah, thank you so much.
No problem. Yes, I helped Miguel. I got him in. He's actually, I don't know where he is right now. I think the last time I talked to him, he is going to be a doctor. So big ups to him. And so I think that he owes me a drink because I got him the internship and got him into med school. So I think this is perfect place. MESA is perfect place to do that, to be able to help each other. Not only figure out what you guys want to do, but also figure out, bring each other in a higher level. I was able to help Miguel get an internship, and that he has never done research before. And so to have that in his plate so that he'll be able to go to med school, I think that's pretty big. Yeah, Jan.
Hey, Ernst.
Yeah.
I wanted to ask more about your unique experience at DVC. So what is one thing that you think is unique and beneficial to your experience at community college that students at a community college won't get at like a UC or a CSU?
I think it's we live really close together. So people in Pleasant Hill, people in Concord, we all live as a community. In four-year college, for example, we drive from faraway places and then come back. And so we don't have that time to be able to connect with some of our peers. With DVC, when I was there in Mesa, I knew almost everyone, and everyone was able to help me when I was struggling with, for example, physics homework or chemistry homework. I think Mitri remembers me writing all the equation in chemistry in the board, fighting with Kazuya or Willie because they didn't think that I was right. And so I don't know if I had that in SFSU. I had a little bit of it, but I had a bigger part that in DVC.
Thank you.
I'm sure Jenna knows about that. Miguel, I think you have a question. Oh yes, let's stop that.
Yeah. So when you were talking about the microtubules, so I don't know much about it, but I've seen videos, and I think I understand that they can form and deform as needed within the cell, right?
Yeah.
So it's interesting to know like that what was it called, that enzyme, the
casein kinase.
casein kinase, without it, like it wouldn't be forming, right? Is that how it works?
Yeah. Well, for us, we don't know 100%, but this is what we're observing that it is. There are other proteins that does confirm that closes up gamma tubulin ring complex. Now, the biggest part of this is that if casein kinase is controlled by, for example, viruses, can the virus control the movement in the cell? Or if a bacteria is in the cell and controls casein kinase, can it control where the microtubule is going to be? And I think that's the main reason why we're studying this, because we don't know.
Oh, yeah, that's interesting.
Yeah.
So because I remember from these videos, just like that the dimers of tubulin go, like they start to form and stack up and start to make up the tubulin. So I thought it was like interactions and what is it called, bonds between like the dimers. But if like this research is suggesting that without the enzyme like at the base of the tubule, the microtubule, they wouldn't be able to like just stack up on them, right?
So what we're learning right now is new. So you won't have this in textbooks, which is the amazing part of research, because we only started to observe this in 2015. That was the latest one that we knew. So in what you're saying also is that in textbook we see that the alpha beta tubulin does stack up to each other, but it doesn't tell you how it's made, or like where it's being made. Is it being made in the cytoskeleton, other cytoskeleton parts, like other microtubules stacking up each other? How does it nucleate? And that's what also interests me in textbook, because they didn't say that. Like they don't give you that answer. So
yeah,
which is amazing.
Thank
you
you
for the question. Angelica said, can you share what your educational pathway experience has been during this COVID-19 pandemic? How do you practice self-care during these times? This is a super good question. Actually, I don't have the best answer, but this is what I did. For my educational pathway, I'm still able to do my master's degree because I'm able to go to labs. And I'm doing part of my lab class in the Krogan lab and do part of it in the Sanchez lab. Now, some people can't do that and they only have one lab, or even the labs that are still closed. So I think I've been lucky, and I'm trying to help other people also get that opportunity, but it is also kind of rare. My self-care is that I walk every day. This is the reason why I live close to UCSF, is because it takes me 20 minutes to walk over there. If I just want to do one thing, like pick up something, I have to walk there 20 minutes and then walk there 20 minutes back. And that walking actually helps me think and just relax and not worry about what's going on. But of course, there are people that's not wearing masks, so it's a wash. Thank you for the question, Angelica.
Hi, Ernst.
Yes, Major.
Can you, I just wanted to share, Ernst has downplayed his challenges here big time. Whatever I have heard from Ernst, or I have known, he had so many challenges, but he came to MESA with a grand smile every day. And he made our lives better with his presence. And we are so grateful that our paths crossed, Ernst. And you deserve every bit of what you have shared today. And look forward to seeing you in many big places. I know you will do us all so proud.
Thank you, Maitri.
Going back, yeah, 3 years, and I see so many of you who will be, who are already in that direction and who are going to be there, because I have interacted with so many of you, and I know you have the potential. But Ernst, one of the common things I think for a lot of our students is internships, right? It's always like, how do I get my first internship? What do I do? Do you want to just share like 3-4 specific points, or even about the Gladstone internship? I don't know if it's still open for this year, but I'm sure all the students here would be interested in finding out how they can make their application portfolio stronger.
So I actually checked it, and they're opening it up again. So it was closed last year. I think moving forward, they are starting to immunize everyone in UCSF and in Gladstone, including myself. I just got my vaccine, and that was really painful, but it got through it. It was pretty cool. So I think they're starting to open up. 3 things I think you need to get a successful internship. Now, this isn't just for the PUMAS program. I think this is all universal to other internship too. In the second year, I actually applied to 10 internships, and 2 of them I got in. One in this UCSF, one with CCMI, and one was in Dogma and college all the way in East Coast. The other 2 I had what's called a wait list. It was UC Merced, and then I think the other one was in Brown University. But all of them had something in common. One is that recommendation is pretty big. I would definitely figure out the best writer that could write you a really good recommendation, because that recommendation will bring you different places. My recommendation came all the way from Nevin Krogan, who owns the Krogan Lab, and he has put me into places because of his recommendation. That's definitely important. Number 2 is to plan ahead. I know a lot of people who don't plan ahead and they just apply, like my friend Miguel. However, planning ahead, what I did is I took all the internships that I know, put them in spreadsheets, and then put a date on it, put out the email, who is going to be the contact for it, put a little description of what you need in that internship. And I do this also with transfers and also in my PhD program. Make that list, and then starting from, what am I interested in, and then highlight what I am interested in. Take out things that I don't want to be, just because it's easy, just because it's, what is it called? Do you want to waste your time in it? Because you're going to have only a certain amount of time writing for different interests or internships. So definitely focus on things that you want to get into, and then diversify that internship. So in, I think 2017 or 2018, I got an internship with JBay, and they do plant biology, which was like, pretty big of me. It's like, wow. It got canceled because our previous president decreases the amount of money that DOE could spend. And so I was one of them, the casualty. But, I think diversifying where you want to do and seeing, okay, not only cell molecular biology, there is microbiology, and, there's a lot of people that are doing a lot of good work in there. And there's a lot of biophysics lab that's doing a lot of good work. And for me, it was easier to get in biophysics because not a lot of people are going to biophysics. But in the same time, the reason why no one's going to biophysics is because it's difficult. Should you stop going to a difficult subject internship just to see if you don't like it? You don't. I mean, that's up to you. But I think it's worth it to go to a difficult internship to see if you'd like it. And so I think those three ones is a start. I think there's other ones we could talk about, but those three ones would start. Have yourself actually a mentor and someone that could recommend you. That's number one. Number two is to plan ahead. And then number three is to diversify. I hope that answers and thank you, Meichi. Those are kind words. It was tough being in DVC and going all through that. But I mean, at the end of the day, I'm enjoying what I'm doing. I'm poised to go to a PhD school next year. And I want to go to UCSF as my PhD. So it's been a road, but it was fun too. Heidi said, did you always want to pursue science? How did you find it? No, I didn't. Actually, in the beginning, I wanted to be a philosophy psychology major. And then I went to DVC. I figured out that maybe not. Then I wanted to be a doctor. Then that was my plan for 2 years. And then Maitri, like I said a while ago, she pushed me to go an internship. And so I went to the internship and I found out like, wow, this is cool. This is, without that internship, I think I would have been a doctor, but I think I wouldn't be happy. This is cool. This is awesome. Like, even though it was a really crappy internship, it was still something that I learned. And that made me say, okay, this is just a stepping stone. I think the next internship is going to be better. And so I pushed myself to get another internship. I planned ahead, I got recommendations, and I got in. And then it just opens the door, because once I finished my internship, they actually stopped me and say, hey, do you want a job? And I was like, is that even a question? I was like, yes, of course I want a job. And so I got a job in the Krogan lab, and now I'm, doing all this amazing things. And so, yeah, that was perseverance right there.
I was wondering if I could say something.
Yeah.
My intention, it's my day off, so I have to do many things with family and whatnot. But my intention was only to be here for about 10, 15 minutes. Oh. But your perseverance was so captivating and your story was so touching that I stuck around and I was just drawn to the screen in your slides and your story. So, wow. I would love to share this recording later on with our students and our awesome program director because it is inspiring. MESA works, it really does. And the networking, the collaboration, and all the resources, you're proof that, yes, perseverance, and just like your slide said, grit, determination, but you're doing it. And look at the time that you're still continuing to do this during COVID-19 and all the restrictions. So I applaud you and right on for sharing your story with us. But now I do need to scat because I have a teenager that's calling my name and he's hungry.
Awesome. Thank you, Angelica.
And it was nice meeting you. And thank you for allowing us to join you during this space. Take care, everybody. Bye-bye.
Awesome. Thank you.
Thank you, Angelica.
Bye, Chandra.
Actually, it would be nice if I could get her email. Maybe we could do a collaboration, do something. It would be cool. So maybe I'll get that from you, Maitri.
Sure.
Or Brittany.
Sure. A quick question. One of the things, I don't know if our students are very quiet here, but one of the questions students often have is that, how do these courses that they do in DVC relate to the internship? I mean, how is that relation? And also sometimes students, they trying very hard, but the courses are very difficult, right? The STEM courses, and they may not get the grade that they have been hoping. Mm-hmm. And so how does that still work? Is it like, I know some students are not good exam takers, for instance, but they're amazing at lab, right? So can you talk a little bit more about this piece?
For me, I know maybe you disagree with this, Meichi, but I wasn't a good student. I was I tried really hard in my courses. I mean, I was also taking care of my son and doing my work. So for me, I found out that a lot of this stuff that you're learning in, for example, chemistry and biology is huge. I know exam sucks and I know homework it's you know it's tough, but knowing some of those actually helped me in my research. For example, the what's called in chemistry, it's C1M1 equals C2M2, and that has been ingrained in me because every time I have to make a solution, every time I have to use my cost thinker, I have to figure out what concentration I have to do with those proteins. If I do too much, then I just killed my sample. And the person who's doing the sample may take like 2 weeks to make that. And so it has to be really good. And so little things like that, like another things like, for example, knowing the central dogma, DNA to RNA to proteins, is something that it's totally ingrained now because I'm working in a proteomics lab. KSHV, for example, doesn't follow that central dogma. It goes from DNA to protein. Just knowing that, that small tidbit actually helped me because now double-stranded DNA to protein, oh yeah, you don't need anything in between. However, exams and homework is definitely hard, and I agree with everyone that it is tough, but I would grind it out. I know sometimes you don't get the grade, but doesn't mean that your grade doesn't mean who you are. You know what I mean? Like, just because I got a C in chemistry doesn't make me a good scientist or a bad scientist. I got a C in chemistry, which is hard, but in the same time, like, I learned a lot from those mistakes. And then once you transfer out from community college, it gets a little bit easier because not only you have more resources in a 4-year school, you'll have people that is you'll have more people in general, and also you'll have tutors that is on hand that and more accessible teachers, so you'll have more opportunity to show your stuff. But grinding it out, finishing your work in the community college, I think is super important. Just remember, your grade doesn't mean who you are.
Can we get the contact information for the club that you're starting? I think it was on a slide, and then
Yeah,
I
will definitely write out. Sorry, I have too much screens. I'll write out the email address. So this club, I am trying to get other colleges into it too, like what was it called, East Bay and Sonoma State. And so I'm hoping to be able to get a good group of people who's doing research in all those areas, and then see if anyone wants to present. I think that's right. Disease Journal Club. Awesome. And we meet every Friday, 4 to 5. I don't want to put pressure, but Jan, who was in this call, was the one who is the person that is going to be presenting. He's a really nice person. As you can see, like, David and Jenna came into my meeting. So these are people that are in these meetings and are able to support other people. Yeah, I think that's the other thing that I want to impose on other people. It's like to be able to be in a community, because like you can see in MESA, you have this community and everybody's able to help each other. once you go to a four-year college, you're going to lose that community and it's going to be really tough. However, if you're still part of a community that is doing science, like for example, hopefully this club is going to be part of it, you'll see that, you know, you'll have help and people are going to be able to push you, not push you, but get you to a better place. You know, again, example with Miguel's part of my friend who, you know, he's also an older student like I do and didn't think that he would get to internship. And then he just asked, and I said, you know, let's work on this and let's grind it out. We made his application. I personally sent that application to the Pumas program, and they knew that I vouched for him, and they said, you know what, we'll take him in, and then he got in. So it's being in that community is important.
Are you open to sharing your email or contact information or a way to connect with you in the future?
Let's use this gmail.com. And just in case, I don't use that, but I know that I might use this more often. Gladstone at ucs.edu.
Ernst, in the remaining few minutes that we have, I was wondering, you know, can you also talk about, you know, this perception that, you know, the UCs are the best place for every student.
Oh,
And
yes.
I totally, I think UCs are great in their own way, but I think CSUs have their own strengths. And so, you know, you're such a great role model for us because you pursued the CSU way and you're doing the, you know, front end research at this point, right?
Mhm.
So do you have anything more to add to it about the CSUs, like why it is such a great place for you?
Right. So there are multiple things in that one. I tell people, number one is that, you know, CSU is number one, it's cheap. I wasn't able to go to UC because of the price tag. You know, it's $16,000. I'm taking care of a child. And on top of that, you know, living in California is ridiculous. And so if price is form factor right there, you know, CSU is a great alternative. And I don't think it's just because it's cheaper doesn't mean it's worse. Number two is that I planned ahead. So this is same thing as what I did with internship. I planned ahead. I wanted to be working in UCSF. Now, if you're looking for other CSU that you know, that doesn't have, there's a, I guess, a research close by. CSU is a great way to be able to go to CSU and do the research in you know, that area. For example, Gladstone is a huge research institute, and I thought that if I go to UCSF, not UCSF, SFSU, I can go work in UCSF, do all the craziness that I've done over there, and then finish my degree in SFSU. And on top of that, I'm still doing crazy things in SFSU. It's not just, you know, like just because I'm in CSU school doesn't mean that we're doing less. We're actually doing as much as you know, as I'm doing in the Krogan lab in UCSF. I showed you guys, you know, the KSHB project that I'm working on. I didn't show you my my plans and all my perspectives, but it's a huge project, and you know, so for someone that's from SFSU, that's pretty big. I think the problem is where it lies is that the amount of how much a school is able to spend on a student. For CSU, yes, it's pretty low. They don't have much money to spend on a student. But if you're if you plan ahead, you're crafty, you're able to figure out, okay, these are the low points of the school, these are the high points of the school. Can I manage the low points like I did? And then you'll be able to get out of CSU. I didn't pay for tuition for 2 years. I haven't paid tuition at all, actually. And I'm going to school for free. And that's you know, that's a blessing, and it's crazy. And I'm able to do what I want to do. And that's because I went to a CSU. I don't know if I would have had the same opportunity in UC. I may have, but I think that either way, I still came out great in SFSU, and people in SFSU have a lot of diversity. It's an amazing school. You know, I meet a lot of people that are amazing. I meet a lot of mentors that are amazing, and who is able to support someone that's first generation, who's a minority. You know, that I think is huge. So I say I tell people, hey, just think about CSU first and see if that's, if you guys don't like it, then, you know, there is the UC that's available, but it's cheaper, and yeah. Sorry, that's, the problem I think is that there are people that think that CSU is kind of lower than UC, and I think that is, it's a bad, what's it called, connotation, because I'm training people from Stanford, people from UC Berkeley, who doesn't know what I'm doing. Does that mean because I went to CSU? I mean, it doesn't make sense to me. And so I think you should do whatever is gives you the most opportunity. And CSU will give you a lot of opportunity. Thanks, guys. Sorry, I ranted for a little bit, just a little bit.
I don't know if this is a good time to ask, but I was wondering if people feel comfortable if they would turn on their cameras so we can take like a screenshot of the event. Anyone that's cool with that?
This is awesome. I get to see people who's listening to my talk.
Yeah. All right. I'm going to give a little countdown. Okay. I guess it's not a countdown. Everyone's going to be like, that's not a countdown. Okay, I'm going to count to 3. And on 3, I'm going to take it. All right. 1, 2, 3. Yeah. And I'll take one more. Okay. 1, 2, 3. All right. Awesome. Thanks, guys.
Awesome. Thanks, you guys, for listening to my
Monica.
presentation. If you guys want to definitely reach out, please reach out. I would love to help anyone that's and you know, see if you guys listen to you guys, you know, going from DVC to wherever you want to transfer to. So please definitely reach out.
Thank you.
No problem. Thank you.